Hi, this is Dr. T, and welcome to my desktop. As you can see, I've got the recording set up behind me for the other videos. Okay, so today I'd like to talk a bit about DNA replication. And to be honest, Wikipedia is another great source. This is going to continue my tour of Wikipedia. Okay, so to talk about DNA replication, so obviously we're doing central dogma, and then we're going to make more DNA. So our cell is probably dividing. It's going to have daughter cells, and it wants to make more DNA so each daughter cell can have it. Okay, so this is actually one of the interesting photos, not really for what it's actually trying to say. So this one's trying to point out that there's certain regions of DNA where this start, and that's entirely true. I mean, that's what the picture's for, but I'm going to go a little higher level. The goal here is that you're going to start out, you're not going to start in replicating all the DNA at the exact same time. Instead, you're going to take your DNA and you're going to have to split it apart, but you're not going to completely separate it. Now, that's what we do when we do polymerase chain reaction. We're going to separate it completely, but that's a very artificial one. And polymerase chain reaction can't handle, at least normally, the whole gigantic chunk of DNA. It likes smaller segments of DNA. Because if you try to take it completely apart, you just get a mess. So what the cells do is they will create this kind of bubble. And this bubble is going to get split apart, and this is where replication is going to take place. These AT-rich sequences, adenosine and thymine both have 2 hydrogen bonds. Cytosine and guanine have 3. So it's easier to split apart an AT-rich sequence than it is to split apart a GC-rich sequence. You're going to split it apart, and you're going to create this bubble. And this bubble is going to kind of be a mirror image. So we're going to take this part of the bubble, or really kind of down here. We're going to bring in a bunch of proteins. We're going to talk more about those here in the next couple of images. But this way, we can grow it this way as I'm tracking over here. And then I can grow it this way as I'm tracking over here. Each one of these is going to become what's called a replication fork, because if you zoom in and we ignore the rest of it, we're going to have a fork. Now we're going to have one fork going one way, one fork going the other way, and we're not just going to do this at one point in the DNA, because it would take too long. You've got 3 billion base pairs to go through. This is going to take forever. Even though we're going through this at insane speeds, we're still going to take a while. So we're going to start in a couple of spots and do this in steps. Okay, so let's zoom into that replication fork. So we're going to get a bunch of proteins brought into it. So this would just be one side of that bubble. Okay, so a lot is going on over here. We get a lot of proteins attaching. So the key one that I'm going to start looking at is this helicase. Enzymes are named ideally. They've got crazy names at times. But ideally, what they do and then end in ace. So these break apart helixes. So it's a helix, the helix, and then ace. It breaks it apart. So a helicase is going to come in here. And by the way, these all actually end up becoming like a gigantic complex. They're not nearly so discrete. But it's going to start splitting apart the helix. Now, this has a couple of interesting applications. One, you'll notice that whenever you take a helix, so if you take a couple of pieces of twine, you wrap them around each other, and then you try and separate them apart by sticking your finger in and zipping it, unzipping it. Well, if it's a normal, if you think about a zipper, they peel apart. But if they're unwinding, as they unwind, they have to coil, they have to spin. So if you're trying to do this, you're now spinning them. And these will actually spin at a rate comparable to, I believe, like a jet engine. It's insane on how fast these go. But obviously, if you keep spinning something and it's not really short, if it's really short, it just kind of flops around. But if it's either attached on the other end, so it's kind of a circle of DNA, and we see those in plasmids, which are small circular DNA segments that bacteria like to exchange. It's kind of like the bacterial version of a flash drive. Or in a person, if we're trying to spin it and it's really long, because keep in mind, your DNA is almost as long as you are tall kind of scenario. Your DNA, if you put it end to end, ends up going like 6 foot. It's ridiculous. That's in every single cell. So what you would end up doing is it's going to create a mess if you try to do that. So you have to unwind it. You have to compensate for that. And that's what this topoisomerase. Topo is going to be the topology Isomer means, kind of the same, and then ace, it changes. So this is keeping the topology the same. This is allowing it to spin. So what these will do, depending on which version you've got, they'll create a nick and allow it to spin, or they'll actually take 2 strands of DNA and pass them back through. But these are going to keep it from becoming knotted and all tangled. So I'd say if you remember the old phone cords, but I know a lot of my students probably don't because those are so long ago. But if you've ever seen the old curly phone cords where you got the phone, it has the dangling one that plugs into the base end because, this is before, cordless phones were a thing. Those all get nangled and gnarled and knotted up, and that's what we're trying to avoid. So isomerase fixes that. Interesting enough, this makes a great drug target. So if you have cells that are dividing really quickly and you want to stop them from doing that, aka kill them, the scenario would be cancer. Then you can target topoisomerases. Slowly growing cells don't need the topoisomerase nearly as much, whereas fast-growing cells are duplicating their RNA. They really need that topoisomerase. And thus, if you can break it, then you can make sure that when it tries to replicate it self-destructs. So that's used quite often on cancer. One of the ways you can target cancer is to go for fast-growing cells. And this is the target. Okay, so you're going to split it apart. You're going to have some proteins just kind of keep everybody split apart. Because remember, these DNA normally has 2 strands. They're sticky. They got hydrogen bonds. They want to do some hydrogen bonds. Not to mention, you've got some hydrophobicity going on here. And it's normally not 2 things you put together. You got some aromatic interactions. They would love to stick back together again. So to prevent that, you've got some proteins that attach. Now, you've got 2 different stories depending on which way you're going. I'm going to start going down here, and then I'll talk about up here. Actually, you know what? I'm going to back up. Let's go to this image. Oh, where is it? Here it is. Okay, so what we are seeing here is this is how you're actually going to start building your nucleic acid. And this actually takes place if it's DNA or an RNA. So you're going to come in and you get your nucleotide. Now your nucleotide has 2 key features to it. It's going to have a 3' end, which is about there. And you have a 5' carbon, which is kind of sticking up. In this picture, it's there. In reality, it's kind of off to the side slightly. The 5' end has a triphosphate. That's going to provide the power to actually do everything you're doing, well, most of what you're doing. So what you're going to do is your polymerase, so it makes a polymer, and it sticks it together, so DNA polymerase. And you'll see that these come in like type 1, type 2, type 3, type 4 things. I don't think there's a type 4. But anyways, so this will then bring this guy in. It sees if the base matches, so T to A, G to C, C to G, that kind of stuff. Sees if the base matches, and then we'll clip off to the phosphates and use that energy to get everything moving along and put that, attach that to the 3' end. So I guess actually the 3' end would be up here. My bad, I have this thing upside down. So the 5' end attaches, of the new one, attaches to the 3' end, the old one. Now, they can go backwards, but they really don't want to. Because if there's a mistake, then it simply chops the one that's wrong off, backs up, and keeps going. If you were to try and go the other way, then you would have to have a whole extra step of recharging it, bringing in the triphosphate. Because if the triphosphate part, if that's wrong, it's a monophosphate once you chop it off, then you'd have to put the phosphates back on. That's extra work. So, what we do is we always grow on the 3' end. The 3' end is where you attach to. So you start with your 5' end, that's the old one, and it grows attaching to the 3' end. So that's the growing end. Okay, so that has a consequence. When I'm looking at my DNA, and these are always hard to see. So I'm growing on my DNA. So this guy, they're running in opposite directions. So it looks like the green is going 5' on this end. That means the green would be a 3' on this end. So, okay, so that way, that means that as I am growing, I'm starting on the 5', and as I go towards the fork of the new strand, then that's going to be on a 3' end. So where my cursor is, I hope y'all can see my cursor, where my cursor is, that should be a 3' end, and that's fine. The polymerase just keeps moving. and adding on, and no big deal. That's fine. But here's the problem. On the other set of DNA, the other strand is going the opposite direction. So now this is the 5' end. That means this is the 3' end of the new one. So the new one is on the top is the green. So as the new one is growing, we get a problem. The DNA polymerase is moving to, in this case, my left, whereas the helicase and everything else is moving to my right. So it needs to do a backstitch. It can't, this DNA polymerase has to go left. It can't go right. So it's going to go left, and it's going to come back and go left, and then come back and go left. It's going to do this kind of backstitch. Now, a little bit extra is going on here. Before you start this, you have an RNA primer, and that allows you to start it. Then you can make your strand, and that backstitching process is going to keep these strands called Osaki fragments. So you put down a bunch of DNA. Obviously, this is zoomed in. You know, it's not just going to be 3 base pairs. But you're going to do an Osaki fragment. And then later on, you're going to have all these little gaps. The RNA primers will be pulled, and then a DNA ligase will go fill in the holes. And that way you get a continuous chain. But you're doing this backstitch. Okay, so that's the core of DNA replication. A couple of applications, though. These RNA primers are going to come back again a little bit later. This does mean when you're trying to do the very ends of your DNA strand, what you're going to run into is a small glitch where all of a sudden it's very hard to copy the very, very end of it because you need some starting material. And this is where telomeres come in. And that's going to be kind of at the end of your chromosomes where you have copy issues, and those are going to become important later. I'll talk about that on another slide. Okay, so yeah, that's DNA replication. I mean, top level overview. Obviously, the article goes into much, much more detail. I'm going to talk more about methylation and that kind of stuff here in a little bit later. So yeah, have a wonderful day.